yours.
Assalamualaikum and a very good morning to everyone. First and foremost, welcome to UKM UMS Oita University first joint EcoHealth Symposium on human T-Lymphotrophic virus type 1 related diseases. So for those who are in the hall and also those who attended online, welcome. So before we start, I'd like to ask everyone to stand up for our national anthem, please. guests can have a seat. Okay, our Honorable Head of Department, Department of Public Health Medicine, Faculty of Medicine, UKM, Professor Dr. Rohaizat. Okay. Our distinguished speakers today, we have Associate Professor Dr. Hidekatsu Iha from Oita University, and Professor Kamarudin Ahmad from BMHRC, UMS. So, Professor Madya, Dr. Azmawati Muhammad Nawi, our moderator for today, and ladies and gentlemen. So, welcome to the symposium today. And to make it short, I think I would like to congratulate all the members of the committee for organizing this symposium. And I would like to thank the funder for today's workshop, which is funded by the small grant from Research Center for Global and Local Infectious Disease, Oita University, Japan. So everyone may be asking about this joint symposium on HTLV-1, and it is mentioned as first, because just to give some introductions that we are actually in the beginning of the process of collaborations, research collaborations on HTLV-1. So we start with the symposium. Thanks to Prof. Iha and Prof. Kamarudin to come up with the idea. And thanks for inviting UKM to be part of this project. So with this symposium, we get to know on HTLV-1, and then also during this program, we are going to have also a series of visits to a few places to see, especially on the field, potential sites for study and so on. So it's going to be a long journey for collaborations, inshallah, through this program. So I think I'm giving the honor to chair the session today and also to introduce to you the moderator for today's sessions. Today's session will be moderated by Associate Professor Dr. Azmawati Muhammad Nawi. She's an Associate Professor in Epidemiology Unit, Department of Public Health Medicine, UKM. Graduated in 2018 for PhD in Molecular Medicine in UMBI. And she has more than 130 plus publications in Scopus and WAS. And her main interest is on NCD, particularly in cancer epidemiology. So just a short introductions on Professor Azmawati. So without further ado, I think I would like to pass the sessions to and welcome Prof. Azmawati to moderate the session. Please welcome.
Assalamualaikum and very good morning. Thank you, Professor Rohaizat, for introducing me. So welcome, thank you and welcomes to the UKM UMS Oita University Eco Health Symposium on basically on the HTLV-1 related disease. So today's session basically we will start with the first speaker, Associate Professor Iha Hidekatsu, and then we have a Q&A sessions. And then after that, we need to a break for a while, tea break. And then we will continue with the second speakers. Okay, bear in your mind, at the end of the two sessions of the speaker, there is a quiz, okay, with 5 giving prizes. So bear in your mind, listen carefully so that you can answer at the end of the sessions. Okay, so let's introduce our first speaker. Associate Professor Iha Hidekatsu was born in 1963 and now at Molecular Oncology Unit, Research Promotion Institute, Department of Microbiology, Faculty of Medicine, Oita University. His education started with Bachelor of Science in 1983 to 1887. Then he continued his study in Master of Science at Kyoto Prefectural University, followed by a PhD in Genetics in 1969 at the Graduate University for Advanced Study in Japan. So for now, Associate Professor as a Unit Leader at Molecular Oncology Unit, Research Promotion Center, and a Chair at Gene Modification Experiment Safety Committee. His career started at University of Yamanashi. He also has been worked at the Lab of Molecular Microbiology, National Institute of Allergy and Infectious Disease in USA. He come back to serve his country in 2004 at Oita University Faculty of Medicine till now. So he is also being associate editor for Frontier of Microbiology. His research interests mainly on the HTLV-1, and part of that is actually a collaborations research with UMS. That's why we are here today. He has published 4 books and more than 50 high-impact journals. So let's listen to his thought on the pathobiology of HTLV-1, associated disease, and molecular machinery of the retrovirus-induced hematologic malignancy and inflammatory disease. The floor is yours.
Thank you, Professor Nai. And I'd like to thank Professor Hassan and Organization Committee for this opportunity to talk about my recent progress in HLV1 research. All right. And my talk title is Pathobiology of HLV1 Associated Diseases, Epidemiology, Molecular Machinery of the Retrovirus. Retrovirus induced hematological malignancies and inflammatory diseases. Before I start, my talk, I'd like to introduce the Oita Prefecture. And most of you know well about the Tokyo or Osaka, but not Oita. Oita Prefecture has it doesn't work. Can I click this? No response. Oh, and it has just 1.1 million people. And talking. All right. Okay. Yeah. Sorry. So from our faculty building, we can see the three mountains: Mount Yufu, Tsurumi, and Takasaki. And at Takasaki, you can find thousands of monkeys, and it is very enjoyable. And then we have lots of hot springs, and in city of Beppu, in wintertime, you can see this steam, coming all over the cities. So you can visit Oita, and I will take you hot springs. All right. And this is the aerial view of Oita University. It's a typical national university in a countryside, and we have medical school with 46 departments, including 6 liberal arts, basic medical science, and clinical science and informatics. Our department is located at the 6th floor of this building, and we have a clinical technologist course and nursing course. The university hospital has more than 600 beds, and many patients around the area will have the diagnosis and treatment here in the hospital. And 2 years ago, we started the new facility, research center for global and local infectious diseases. And we have 4 research departments, and I belong to a pathophysiology division, and there are several studies for the infectious disease, including H. pylori. It's conducted by a world-famous Dr. Yamaoka, and multiple drug-resistant bacteria, rabies, and gastroenteritis viruses, like Professor Ahmed conducted in UMS. And also, I do research here on HLV-1. So today's topics are divided into 3 parts. And first, I would like to introduce the basic of the virus and retrovirus, because some of you, I think I heard some of you are not really familiar with the virus or retrovirus. Okay, so let me start with the virus study. As you may know, virus is very small, and the size is expand between 10 to 100 nanometer diameter. So, and if you put this 20-meter gas tank as a human cell, virus is like a variable, and you can tell how small virus is. The diameter is 100, and so volume is 1 million smaller than the human cell. And for HLV-1, we have, it has very characteristic feature, especially it has the unique reverse transcriptase. So what do we call retrovirus is as you can see here, reverse transcriptase containing oncogenic virus is the origin of the name. So it has plus strand RNA. It's one pair of plus strand RNA and surrounded by a nuclear capsid. And capsid has icosahedral room. Then inside the capsid, we have functional proteins like reverse transcriptase, protease, and integrase. And once retrovirus create the double-strand DNA from the single-strand RNA, integrase lead this provirus, double-strand DNA provirus lead into nucleus, and the integrase put the nick on the host genome DNA, and the irreversible infection is established. So this is the small cartoon how retrovirus infect and get into the nucleus. Oh, no. It doesn't work. All right, okay. All right. So it doesn't work, sorry. And many of you know, about HIV, but you are not really familiar with HTLV-1. Actually, they are very close, virus. The common feature of this virus are, having basically same component in their genome, and reverse transcriptase and also integrase. So it's like same virus, but the outcome of the infection is totally different. In the case of HIV, as you all know, if you don't do anything, you have almost 100% AIDS symptom after 10 or 20 years of the infection. In the case of HTLV-1, it has around 5% of the T-cell malignancy, and rest of them are not has that a leukemia or lymphoma. All right. So they are both tropic to or CD4 helper T cell, but the outcome of symptom are totally different. All right. So from now, from here, I'd like to talk about the HTLV-1 related diseases a little bit more. The first one is adult T-cell leukemia. HTLV-1 transmit either sexual intercourse and mother-to-child transmission through breast milk feeding. So in the breast milk from the infected individual, you have 1 million virus in 1 milliliter breast milk, and it goes down to the intestine and through the pile patch, HTLV-1 infect to a child. Once the infection established, you have to deal with this virus lifetime, right? And the malignancy is caused by two oncogenic protein called TAX and HBZ. TAX has, as you can see here, has the activity for persistent NF-kappa B activation, and also ability to induce genetic and epigenetic mutation to the host cell. HBZ, on the other hand, it has immune suppressive feature. And many of you know the patient of certain malignancy, they have very, inactive immune system. That's why malignant cell propagate very easily. In the case of ATL, HBZ play a crucial role for this onset of ATL. Besides this malignancy, HTLV-1 cause some neurological inflammation. One is caused by HTLV-1 associated myelopathy. The HTLV-1 positive T cell infiltrate everywhere in your body. And when it goes to spinal cord, immune cell attack this site, then the individual has dysuria, dysphagia, and the worst one is dysplasia. So many individuals cannot even walk after the onset of inflammation. Okay.
Saya just suruh komen dulu dekat saya untuk kongsi.
Okay, right. So two inflammatory diseases. And I'd like to show one really important information. For these two inflammatory diseases, especially HAM. Okay, in the next slide, I wanted to show you the video which is announcing the establishment of the new national center for neuronal rehabilitation in Malaysia. We are collaborating with the Japanese venture company Cyberdyne. Number 12. Yes. The next one. All right, yeah. And you can see the robot here. It helps the disabled person, becoming, walking by their own. And I can show you the video how it is working.
can damage this network loop, resulting in difficulties moving. Even for a person with a disability, signals sent from the brain to the muscles leaks onto the skin surface as a very faint signal. We installed innovative sensors on HAL, which could read and analyze the signal so that the robot could be controlled voluntarily by the wearer's intention. What's important is the neural loop between the brain and the other parts of the body. Repeated exchange of the signals will reinforce the synaptic connections and drive relearning. Cybernetic treatment improves and regenerates the physical function of the patients who suffer from disease, such as neuromuscular disease.
We treated now about 100 patients with spinal cord injuries with the HAL robot suit system. And we see dramatic changes in their ability to move again. We see in some patients also some improvement of their pain. We see improvements of their sensory, which is very important to prevent pressure ulcers. And in some patients, we also see changes in their urinary function.
All right, and I believe once this National Institute start their operation, Malaysia will be the leading country for neurorehabilitation science. All right, so let's go back to ATL. So this virus transmits through the mother breast milk. So HLV-1 infect into a CD4 T cell. And once the infection is established in a baby's body, first oncogene TAGS has the activity for immortalization, but it is not the true cancer cell. You need multiple mutations. And finally, second oncogene, HBZ, it induces the FOXP3 and Treg phenotype. Highly immune suppressive cell develop ATL cell. In the case of our country, Japan, the average onset of this leukemia is 68. But in the case of developing country, it starts as early as 30s or 40s. So once it started, it's very hard to cure. So TAGS induces immortalization and genetic and epigenetic mutations. SBZ, immune suppression, and onset of ATL. So many researchers want to find out the true biological marker for the risk of ATL onset. But so far, it is not really successful. So the best way to avoid this disease is prevention. In our country, the pregnant individual, 100% of pregnant individual has to take the examination of this virus infection. And the population of the HCV positive people in Japan rapidly decreasing. And Professor Ahmed talked later about the prevalence of this virus in this country. And we can start this, prevention program here in this country. All right. And it is the comparison of the two viruses, HLV-1 and HIV. So you may, many of you know, the virus, retrovirus transcription happen to the left side of the, we call it long terminal repeat, LTR. And the transcription, the direction is one way, usually. And tax oncogene indicated in red box transcribed from the left LTR. But in the case of HBZ, the transcription initiate the opposite side of the provirus. So it's mutually exclusive genes between tax and HBC. So, sorry, okay. All right, sorry. So first, tax works as, for the T-cell immobilization. And then it also induces epigenetic and genetic mutation. It eventually downregulates its own expression. Then after that, HBZ activated and do the last job for ATL onset. All right? And what's the nature of this T-cell malignancy is coming from the regulatory T-cell feature. As you may know, we have 3 different T-cells. One is helper T-cell, many helper T-cell, Th1, Th2, TFH, and others. And another one is effector cell, CD8. It can kill virally infected cell and also tumors. The last one is regulatory T cell. It has CD4 and CD25 for its own cellular surface. What the role of regulatory T cell is? It will stop the immune reaction caused by those, CD4 and CD8, because you cannot continue immune reaction forever. You have to stop at a certain point. Regulatory T cell does this job. But in the case of HCTM infected cell, it eventually, induces HBZ. HBZ does multiple job for many cellular proteins, and eventually it has regulatory T cell feature, and you cannot control anymore. It expands until the patient, dies. So it's very difficult disease. HL cell, like inducible Treg, so it downregulates old immune reaction, especially Th1 reaction. It works for anti-tumor and antiviral activity. So the first part, I explained how HCTM does bad thing to our body, and TAX and HBZ are 2 key players to lead the viral infected cell to malignant, very severe malignant cell. So at the beginning, clinician try to, it jumped. Okay, yeah, no, not here. So this virus discovered 1981, and then people knows, it caused the very incurable leukemia and lymphoma. And they start with the standard chemotherapy to the patient. And this Kaplan-Meier figure is the first report how this disease is difficult to cure. Professor Shimoyama conducted the research on the more than 100 HTLV patients, and then he noticed there are 4 different pathogenic types. The worst one is acute, and same lymphomatous, and chronic, just a little bit better, and smoldering. So when you take a look at the median survival time, it is very, very short. More than half of the HTLV patient die before a year, and only 20% has a 3-year survival period. And from this research, Dr. Shimoyama found the prognostic marker like white blood cell count, soluble IL-2 receptor, and LDH. They start with this standard procedure for the hematological malignancy with CHOP, and the 2-year survival is just 15%. And then they developed better treatment, modified leukemia study group 15, LSG 15, and it improved the survival rate from 15 to 24. And also it's for a 3-year survival. But this treatment has very severe side effect, and you have to stay in at least 6 months in a hospital. So especially elderly people doesn't want to this therapy, and they take standard CHOP. Even the survival period will be shortened because you can be an outpatient in this therapy. Okay. Then they started to hematopoietic stem cell transplantation, and it worked better than chemical treatment. It has 40% of the 5-year survival rate. But as you take a closer look at these numbers, rest of them, 60% of the patient die from severe, very severe GVHD, graft versus host disease, or just the progression of ATL. So only 40% can be saved this treatment. And it is also a problem because it's, it is transplantation. It's very hard to find donor and recipient. All right. For a long time, chemotherapy and hematological transplantation were only two methods for the treatment of HTLV. But in France, the French group started this antiviral therapy. AZT is the first chemical to cure HIV. It's an antiviral treatment, and interferon also antiviral treatment. And what they got is very fantastic outcome. For the patient with this antiviral treatment, patient has nearly 50% of 5-year survival. And chemotherapy just 12 months. So it's really nice. But it also has pitfall. When it comes to acute type, it has much better outcome than chemotherapy. But if you take a look at lymphomatous type, there's no effect. So it works only for leukemia. It doesn't work lymphoma. So third therapy, nice, but still, we need more tool to cure this disease. So last 10 years, we had 4 novel treatments method for ATL. The first one is antibody therapy. As I showed the previous slide, ATL cell behave like Treg, and true Treg has many markers on their surface, and CCR4 is one of the most predominant Treg cellular surface protein. So targeting this protein, you can expect 2 curable effects. One is direct antibody-direct cytotoxic activity with this antibody. Another one is because CCR4 is basically expressed in a highly active Treg, you can decrease immune suppressive Treg with this treatment and reactivate the immune system in your body. And then it attacks ATL cell. This treatment conducted in phase 2 experiment, and median progression-free survival is not like this, but overall survival period is like chemotherapy. And actually, this requires just 1 shot of this antibody. So it is a good alternative for bone marrow transplantation or chemotherapy. And if it works, it works very well. And you can see the picture, the infiltrated tumor cell on the surface of the hands, it disappears. It's complete remission. Okay, so antibody therapy is one of the very powerful tools. Okay, and it is approved 10 years ago or 12 years ago. Okay, but the mogamulizumab therapy has side effect, and the most severe one is it induces high acute activation of immune system, and it leads to dermatological information, including Stevens-Johnson syndrome. So you have to be very careful to give this shot. And also you have to do a pre-diagnostic check before you give this antibody. The second one is CD30 antibody conjugated with the toxic material. As I showed, CCR4 is a predominant marker of the Treg. Another one, CD30, is also a good marker for Treg. So pharmaceutical company conjugated Vedotin. It's a microtubule inhibitor. And with CD30 antibody, and CD30 deliver this microtubule inhibitor into the CD30 positive cell. And most ATL cell express CD30 well. So Vedotin bring this inhibitor into the ATL cell. And apoptosis happen. And phase 3 experiment is like this. It's a little bit better than the mogamulizumab. It's 16 months survival. And it was approved. The third one is the epigenetic inhibitor, histone deacetylase inhibitor. Histone deacetylase is activate many different tumor cells, and it facilitates tumor cell growth by modulating or suppressing anti-tumor genes like p53 and other anti-oncogenes. So once you broke HDAC, you can induce this anti-oncogene and induces the apoptosis to ATL cell. And phase 2 and phase 3 experiment for this, it's okay, but progression-free survival is just less than 2 months. And overall survival rate is also 8 months. You can use it if you don't have any tool for cure, but it's not really impressive. So I showed conventional treatment and also a novel treatment for ATL therapy. And from now, I show the immunomodulator that I, took a important part for this, the machinery of the efficacy. And sorry, but you can read this gene paper, and we listed all the anti-ATL therapy in this paper. Okay, so last month we just published this immunomodulatory drug use for ATL therapy in a Frontier journal. So most of you know, not really familiar with the immunomodulatory drugs, IMIDs, and it has this, the two characteristic structure. the structure surrounded by the red square, this part is IMID. And then it binds to E3 enzyme, cerebrum, and modulate the target specificity of this E3 enzyme. Once you treat this chemical, it binds to cerebrum and induces the degradation of the proteins listed in here, Iros, Icaros, and CK1 alpha. Iros and Icaros also called as IKZF3 and IKZF1. And they are degraded by, through the UV extrusion and also proteasomal degradation. Eventually, if you lose, in a tumor cell, if you lose IKZF1 and 3, you cannot activate IR4 and downstream MYC. You know, MYC is one of the Yamanaka factor. It induces the very strong cell growth, cell cycle progression machinery. So it is very important for the tumorigenesis. Lenalidomide downregulate IRF4 and MYC through cerebrum. Okay, and to degrade the IKZF1 and 3. IKZF1 and 3 in the B cell, it is very important for the differentiation of B cell. As you can see in this figure, red line and, okay, 10 minutes, yeah. It upregulated as the cell differentiation goes. On the other hand, IKZF2 downregulated, and this IKZF1, 3 activation and downregulation of IKZF2 is required for B cell differentiation, but it is also very important to keep multiple myeloma cell alive. So through lenalidomide treatment and downregulation of IKZF1, 3, you lose the promoter of this, malignant cell growth. You can kill multiple myeloma cell. Okay, but it's the case of B cell malignancy. So in the HTL cell, actually, it has not only IKZF1, 3, and also a very strong IKZF2 expression in HTL cell. That's why it is very hard to kill HTL cell through lenalidomide. And so it is a bit complicated, but I generated lenalidomide resistant multiple myeloma cell from the lenalidomide sensitive cell. And after establishment of resistant variant, I found the resistant tumor cell induces 11 times more IKZF2 expression. So in this case, refractory multiple myeloma case and also ATL case, if you have strong IKZF2 expression, it's very hard to cure the ATL, right? But the two clinical study showed very good overall survival rate. It's the best value among the newly developed ATL treatment. It has 20 more months survival rate. They found this good result, but they don't know how this good result achieved. So I was asked from the pharmaceutical company to figure out the molecular machinery of the efficacy. So I started to read the literature, and I found one very important paper in, after 1989 whole exon sequencing of the ATL patient genome, IKZF deletion happened among 15 patients. So in such a case, the IKZF deleted patient, if you give lenalidomide, you can downregulate all the IKZF1 to 3, and it may, cause the efficacy to this patient. And from that publication, I start with 13 ATL cell line, how they behave after lenalidomide treatment. And I found 2 lenalidomide sensitive cell lines at 102 and TL1. After treatment of lenalidomide, okay, you can see the downregulation of IKZF1 and 3 is natural because lenalidomide induces these 2 protein degradation. But between IKZF1 and 3, you can see in the case of HAT102, this cell lose IKZF2 from the beginning, and sensitive TL1 reduces the IKZF2 in unknown machinery. So if you can kill all the IKZF protein, you can kill ATL cell and downregulate tumor promoter IRF4 and CMYC. Okay, so I thought IKZF2 does something for the resistance of the lenalidomide treatment. And we downregulate, knock down IKZF2 like this. for lenalidomide resistant HL cell. After a knockdown, we can reduce the survival rate of this resistant cell. So IKZF2 is crucial for lenalidomide resistance. And we did reverse experiment. We knock down cerebrum. It's an E3 enzyme to knock down IKZF1 and 3. H102 is the sensitive to lenalidomide in basic nature. But after knocking down CRBN, you don't have E3 enzyme. So you cannot kill cell even after a high dose of lenalidomide. And IL-4 and c-MYC induced. It's time? Okay, sorry. So the microarray analysis also support this machinery, and mice model also support our idea. And this is the last one. Even though lenalidomide resistant cell, HL cell, if you use iberdomide, you can downregulate all the malignant protein, as you can see in this square area. All right, so lenalidomide or IMIS induces HL cell death through downregulation of all the IKZF molecules, and it eventually downregulate IL-4 and MYC. It induces apoptosis of HL cell. Okay, so you can read our publication if you want to know the detail. So I will stop here and give the next talk to Professor Ahmed.
Thank you very much, Professor Iha Hidekatsu, for your sharing about this HTL1. Maybe some of you is very new about these topics, but we learn together. Is there any questions? We will allow only maybe one questions. Is there any question that you want to ask? Any questions? Any questions that anyone to ask? Maybe I can start first with what do you think about Professor? What do you think about gene editing in this HTL1 disease related?
For treatment?
Yeah.
Yeah, you can try, but in a real life, we don't have this, method for this treatment. So if you want to apply it, we have to prove the gene editing effect with in cellular level and in vivo with experimental animal. And if you can get the nice result, you can expand to a clinical setting. But right now, I don't think people start this. Yeah.
Okay. Okay. So I think there's no questions. We also have a very short time. So thank you very much for Professor Dr. Iha Hidekatsu for his sharing. Thank you. Thank you. So basically, after this, we will have a tea break, 30 minutes tea break. And for the MMA CPD, we will display at the end of our second sessions. So I will see you later at 11 a.m. after you are having your tea break. Thank you.
Check 1, 2, 2.
Cubaan suara 1, 2, 3.
Check.
Okay, welcome back. We will continue our second sessions with Professor Dr. Kamaluddin Ahmad. So let me introduce first. Professor Dr. Kamaluddin Ahmad was born in 1962 in Dhaka. He is Japanese citizen, so he can speak Japanese. His education started with MBBS from University of Dhaka in 1986, DTM from the Institute of Tropical Medicine, Nagasaki University in 1988, and his PhD from Nagasaki University in 1992. He's teaching mainly on microbiology, immunology, and infectious disease to the medical students, infectious disease to the master in tropical medicine students, biology, microbiology, immunology to science student, and epidemiology of tropical disease to doctor of public health. He has supervised 7 PhD students, 9 master students, and 37 undergraduate students. His research field mainly on molecular epidemiology, emerging infectious disease, and zoonosis, as well as diagnostic to infectious disease. He has published more than 150 high-impact articles and 6 chapters in the books. He also is visiting professor at Nagasaki University from December 2019 and Oita University, Japan from July 2022. He also been an editor-in-chief of Borneo Journal of Medical Science in many editors of international journals such as PLOS ONE, Vaccines, and etc. So let's welcome and hear Prof. Kamaluddin with a topic on serosurveillance of HTLV-1 in Sabah. The floor is yours, Professor. Thank you.
Thank you, Prof, for the introduction. First of all, I would like to thank Prof. Rohaizat and others who kindly arranged this seminar for us. We start from here. The seroprevalence of HTLV-1 in Sabah. HTLV we can divide into actually 4 types. One is HTLV-1 and 2, which are pathogenic for human and prevalent worldwide, I wrote. And HTLV-3 and 4, these are primate HTLV. So HTLV-1 was first oncogenic human retrovirus discovered in 1980. It's my honor and privilege that it was first discovered in Nagasaki University by Professor Hino, right? And subsequently, many research was also done in Nagasaki University. I mean, how it transferred from through breast milk to babies. This was also found in Nagasaki University. The story goes like this. I was not actually a HTLV person. Prof. Iha dragged me into this field to study. In 2016, when I came to Sabah, University Malaysia, Sabah, Prof. Iha asked me to start collaboration in this field. The reason I wrote down here, why Sabah? Why not West Malaysia? The things goes like this. Sorry. Okay, but by the time he fixed the slides, I tell you the story. Why Sabah? In one district of Sabah is called Sandakan, if you know. Sandakan has a very long history of relationship with Japan, trading. About 400 years history of trading between Sandakan and southern part of Japan. So that was one of the reasons why we wanted to do HTLV-1. Because in Japanese literature, I mean olden literature, it said that HTLV-1 came from the south. So south it may be any islands in the south, but we targeted Sabah, Borneo as a whole, but mainly in Sabah. The other reason was, you know, Japanese army occupied Sabah for a long period of time. So we thought maybe that is one of the reasons. The other reason is Tawau area of Sabah. Many Japanese stayed there for a long period of time. If you go there, there are roads named according to the Japanese name, Jalan Kubota or something like this. So therefore, that's the reason why we targeted doing HTLV research in Sabah. Okay. I told you Nagasaki is the one of the Nagasaki University is one of the leading university in Japan, deal with HTLV-1. When I went there first, my professor asked me about cannot control. Okay, maybe this is fine. So if you use, nowadays, most of the diagnosis is done by PCR or other method. In the olden days, they used to diagnose by microscopy. So this is the typical cell or lymphocytes found in HTLV-1 leukemic cell. It's called flower cells. So human T-Lymphotrophic virus, I am sure Prof has said, 10 million people globally infected, but we are lucky 90% of these carrier, they don't exhibit any symptoms. And those remaining people, that's 5 to 10%, infected may develop leukemia or a progressive paralytic disease. The virus changes the folding pattern of DNA. That's why, and there are several mutations. That's why the morphology of the nucleus became this flower shape. The disease HTLV-1 cause, we can divide mainly into two. One is malignancy and other is inflammatory syndrome. So in malignancy, we have adult T-cell leukemia. This is the most common. The other is cutaneous T lymphoma. Among the inflammatory syndrome, the most common we call is HAM, HTLV-1 associated myelopathy, or it's another old name was tropical spastic paraparesis. We have uveitis, arthropathy, Sjögren syndrome, polymyositis, thyroiditis, pneumopathy, and T lymphocyte alveolitis. So you can see a lot of disease can happen if a person is infected with HTLV-1 virus. Therefore, the symptoms also varies. Atypical symptoms occur. Person may complain of low back pain. They may complain of constipation, bladder dysfunction, or muscle weakness. So different form of symptoms appear for HTLV-1 infection. This is the distribution of HTLV-1. The prevalence varies from very low to high. You can see the slide is not very clear, but as a whole, you can identify that in many parts of the world, we have HTLV-1 infection, mainly in eastern side of Asia and western part of Asia, some European countries, African countries, South American countries, and the United States. So highly endemic HTLV-1 countries are Japan, Caribbean region, South America, tropical Africa, Australia, and Melanesia. The mode of transmission, mainly it's passed from mother to child through breastfeeding. And sexually transmitted mission occur, but it's not the major. Major is mother to child transmission. From blood transfusion, it may occur, and those people who are sharing needle by any form, tattooing or drug abuse, it can be transmitted. The genotypes recognized all over the world are 7. Type A, mainly found many parts of the world, and type B, D, E, F, G, found in Central Africa, and type C, it's only found in Asia. And that's Dr. Iha's main interest, depending on the epidemiology. If you see this slide, on the right side, genotype A can be divided into different subgenotype. There is Japanese subgroup, West African subgroup, North African subgroup, different type. But if you see the genotype C, it's actually came from some part of Asia and passed through Borneo and then to Melanesia, Aborigine African, Aborigine Australians and Papua New Guinea. So therefore, our thinking is possibly it passed through possibly West Malaysia or mainly through Borneo Island and passed to the Australia and Papua New Guinea. If you see the epidemiology of HTLV-1 in Southeast Asia, in very few countries, studies has been done. In Indonesia, they find out HTLV-1 and HTLV-2 mainly in intravenous drug abuser, those men who have sex with men. But in Irian Jaya, other part of Papua New Guinea, there are sporadic cases of HTLV-1 in isolated tribes. HTLV-1 is present in the general population of the Philippines, but HTLV-1 is absent among the Thai population, but has been found in prostitutes who served Japanese tourists. And in Malaysia, when I extensively searched, I find out one paper published in Malaysian Journal of Pathology. It's a of one patient of Indian origin, Malaysian, of course. The root could not be identified, but the disease was confirmed by PCR. And there is one thesis, it was never published, at least I did not find, a prevalence. The samples were 140 samples were taken from adults with lymphoid leukemia, myeloid leukemia, and lymphoma. And among them, about 9.3% were positive for HTLV-1 virus. So it indicates there is HTLV-1 in West Malaysia as well. I told you already why we choose Sabah first for our study. Sandakan had a close relation historically with Japan. And then Japanese army occupied Sabah for 3 years. and the ancestors of HTLV-1 possibly passed through Sabah and went to Indonesia or Papua New Guinea or Australia. Therefore, we did this study among the population of Sabah to find out the situation. We can divide our study into three phases. Phase 1 study, we collected blood serum from blood donors of Queen Elizabeth II. We have two hospitals, Queen Elizabeth Hospital and the other one is Queen Elizabeth Hospital II. There is our Sabah's main blood transfusion department. So we obtained the ethical clearance from the Ministry of Health. The second phase study was among the urban sanitary worker of Kota Kinabalu City Hall, City Office. We obtained the ethical clearance from UMS. And the third phase study is among the rural communities from Kudat district. It is north of Sabah. Ethical clearance was obtained from the Ministry of Health. The span of study is something like this. 2016 to 2018, we collected the blood from Queen Elizabeth II. DBKK, that's Kota Kinabalu City Hall, we collected in 2017. The third phase sample were actually collected. This is a retrospective sample. It was collected and stored in our freezer. It was collected in 2014 and 2015. Why the study is like this, it will be explained in the next slide. Before that, I tell you, we used this serum for detection of HTLV-1. HTLV-1 detection has a two-phase detection system. One is first-line detection. We use a commercial kit, which is called Serodia HTLV-1. It's a passive particle agglutination test kit. On the right side, you can see we can, these are all the unsanitized particle as a control we give. Then we give the sensitized particle, and we see whether the agglutination is there or not. This type of agglutination is very clear. We say them positive. These are also very clear. We call them negative, similar with this one. There is another, what we call plus minus, inconclusive. It's something in between the positive and the negative reaction. There is a circle surrounding the positive like pattern. So this is the result. We collected 1,176 samples from the blood donor. The male-female ratio was male were more, 3.7 to 1, male donating the blood more possibly. Median age was 32, and the ethnic group was 33% Malay. Kadazan ethnicity was 15%, then Chinese, Bajau, Dusun, and other ethnic groups. From the urban sanitary worker, we collected 304 samples. Again, male was dominant because of the working thing. Median age was 42. In this group of sample, Dusun ethnicity was main, 32.2%, and Kadazan was 19.7%. So more than 50% of them belong to Kadazan Dusun community, then Brunayan, Bajau, Rungus, and other group. In rural community sample from Kudat area, we collected 1,864 sample. The male-female ratio, it's fine. Female little bit more, 1:1.2. The median age was very young, 25 years. Ethnicity was not recorded. As I told you, this was a retrospective sample. Their idea was different than our idea. But we know from that in Kudat area, mainly the Rungs ethnicity are there. So this is the result. Sorry. Healthy blood donors from Queen Elizabeth, all were negative. Urban sanitary worker from Kota Kinabalu city, all were negative. In the rural community of Kudat, we find out positive 45%. Positive means agglutination test positive, and inconclusive, that's plus minus, was 60. Negative was 1,759. So this is the second line of confirmatory diagnosis. It's a line immunoassay. The commercial name is Inolia test. It's for HTLV-1 and 2. So you can see these are all control if it come positive. The positive reaction is if P19 or GP46, one of them become positive, or GP19 or GP21, sorry, P19 or GP21 become positive, it's also positive. If two like this become positive, it's also taken as positive. So there are some criteria which are written here. If it become positive, we can take as positive reaction. The lower part is to confirm whether it's HTLV-1 or HTLV-2. So we find out among Kudat sample, 64 were positive by this line immunoassay. That's 3.4% was positive. The male-female ratio was, we were surprised because male were dominant. If for every one female, 2 were positive in this case. The median age of the positive HTLV-1 people were 32 years. We don't know why male was dominant, but we tried to do some statistical analysis. And the age also, we try to find out whether the age has any effect or not. You can see when we arrange the age group, this is the control, I mean, all sample group, and this is the HTLV-1 positive group. You can see 40-49 age group were more in the positive group. So we did chi-square test and it became significant. So we can say that there were some relationship with the category and the status of HTLV-1 and age. For the gender, as I told you, the upper part shows gender distribution for all samples, and the lower one shows for HTLV-1 positive. When we did chi-square and logistic regression, both shows there is a significant relationship for male dominance. So every males were 2 times more likely to have HTLV-1 than female. We also tried to see whether there is any geographical distribution of these HTLV-1 positive cases. If I explain you clearly, this is map of Sabah. This region is actually, this is Pitas, this is Kudat, and the island also belong to Kudat. The name of the island is Bangi Island. So if you see the distribution of the black indicate the negative cases and the red indicate the positive cases. We did not find out any special distribution of the cases, but we find some cluster of cases, that's all, but possibly these were not very significant. So this is the current prevalence map of HTLV-1. You can see this is we propose as the new one. Sabah is indicated here. So this is the timeline in future. We hope this is only an arbitrary thing. We don't know whether we can do, but this is our hope. We want to explore new areas in Malaysia. That's West Malaysia, other part of Sabah. We want to explore among the lymphoma patient. As I showed you the paper and the PhD thesis also. We can organize seminars for clinicians, policymakers, researchers. We can do the genotyping of Malaysian strains, whether they are different between two parts of Malaysia or same. We can do evolutionary research, research on non-human primate, and so on. So that's our future target. The study was done by many collaborators from Oita University, Kyushu University, London School, National Institute of Infectious Disease, Japan, University of Glasgow, San Mariana University, Japan, Infectious Disease Society, Sabah, and Menzies School of Health Research, Health Research, Clinical Research Unit, Queen Elizabeth Hospital 2, University Technology Mara, and University Malaysia, Sabah. And the study was supported by different grants. And this is my contact. Thank you very much.
Thank you very much, Professor Kamarudin. Now open for the Q&A session. Is there any questions for Professor?
Can I ask a question?
Yes, please.
I'm Dr. Noraida. Can you hear me? I'm online.
Yes, we can hear you. Please
Yeah,
ask.
I'm a hematopathologist from UKM, and I do a lot of lymphoma diagnosis. And well, you know that we do not screen. I do not know the HTLV-1 status of the patients. And the thing is that when we look at the T-cell lymphoma, adult T-cell lymphoma may look like other types of T-cell lymphoma, like peripheral T-cell, anaplastic large cell, and also Hodgkin. So we do not know the status of our patient, whether they are HTLV-1 positive. I would like to ask Prof. Iha, when do they screen the patient for the HTLV-1 status? Is it when they know the patient has a T-cell lymphoma, or they do general screening?
So
Thank you.
your questions to Prof. Iha, is it?
Yes, because in Malaysia, we do not know the status of the HTLV-1 in the patient who has T-cell lymphoma. So, but in Japan, when do they know the status of the patient's HTLV-1? Is it when the patient has T-cell lymphoma, or they do general screening on the population? Is that clear?
Prof. Iha?
Yes, usually the patient come to the hospital with very how to say fatigue, and because they think they have other disease, but after the test, they found the HCTM infection, and finally it is diagnosed as a this kind of case frequently observed.
Sorry, Prof. Kamarudin, I cannot hear the answer. Perhaps Prof.
It's not
Kamarudin, could
really
you?
clear. Sorry, Prof. It's not really clear. Can you say it again?
I think the question was whether it's already diagnosed previously or newly diagnosed. In many cases in Japan, for example, for antenatal checkup, it's a must for all ladies, not men,
Oh.
ladies to check when they are pregnant. So the status of the newborn and the mother is known, whether they have HTLV-1 or not. For other cases, for unknown cases, mainly in men, adult men, it's when they come, they have to do tests in the hospital. And if it is lymphoma, they definitely go for diagnosis. Not only lymphoma, I showed that there are inflammatory diseases as well. And in neurological disease also, the physician is aware of HTLV-1, therefore they go for the testing. Is that okay?
Yes, thank you. If I have a case from Kudat, I probably ask you to check the HTLV-1 status then.
Sure.
If I get a lymphoma case from Kudat, then maybe we should check their HTLV-1 status.
Maybe you can send the serum to us because we have the agglutination test kit. We can look for it.
Yes, yes, sure. Thank you, Prof. Kamarudin.
Thank you very much.
Okay, thank you. There's one questions coming from the chat. Can we open? Chat board, right? There's one questions. So the question is about your research question on the expectation if the same study conducted in West Malaysia, how it can be linked to the East Malaysia or Japan. It's very difficult.
That's I mentioned the thing, so maybe I have to answer the thing as well. I think the question will be a little bit different. Not with Japanese invasion, but the path we describe, how it evolved and it came to this archipelago, Malaysia, Indonesia, and then went to Australia and Papua New Guinea. Maybe that will be the research question. I mean, the pathway. Because if you see the evolution of HTLV-1, it was primate HTLV first, and then it jumped from primate to human. And this jumping occurs, they say, somewhere near Indonesia. It's not sure. It can be in Malaysia. It can be in Indonesia. Therefore, I think the pathway, how it transmitted in the olden days during the evolution, that will be the research question. And in Malaysia, Indonesia, people are intermingling a lot. I mean, interracial marriage is frequent. So that can be another question.
Yep. Any other questions? Anyone here? Yes, please.
Not area. And the second one, if we look through at the Kudat district area, you mentioned there was prominent of the Rungus races there. But if you look through at the Bangi Island itself, there will be a lot of Philippines area, which is non-Malaysians. Does it include in these samples? That's all. Thank you.
You want a frank reply? I will not hide. You can see the study period. First, when we started, we had grant and we could do the study. So we started in Kota Kinabalu, thinking that it's the capital city. So many people come from different parts of Sabah, and it will be an easier approach to find out whether it's prevalent in other part of Sabah. therefore, we started our study in Kota Kinabalu. But you see the distribution of ethnic group. It's not the Rungus, it's the Malay who donated the blood mainly. If we can see the slides, yeah, I remember. It was the Malay and other ethnic groups, Chinese also, they donated, Kadazan as well. And therefore, when we get all negative sample, he is the grant bringer, so he was very afraid. I mean, how you can reply to, how you can answer the granting agency, because you cannot report negative result, right? But it was good for Sabahan, because they did not have it. Then we went for Kota Kinabalu city office, thinking the same way, that possibly other ethnic groups will be there dominating and we can find positive. But we did not. Therefore, this was unintentional, Kudat. You can say we are lucky because the sample was there already collected. It's a retrospective sample and we got it. Our target was to go for Sandakan, to go for Tawau. Now we find out positive result, possibly we can get grant, and now we will go for Sandakan and Tawau, and as well as West Malaysia or other part, if the grant become big.
Yeah. Okay. Any other questions? Yes.
In Japan, they do the antenatal screening. How do they counsel the mothers who are screen positive? And what is the next step of action? And as for the study in Malaysia, for the serum, because you said it was retrospective, so the patients who were positive for the HTLV-1, what happens next for them?
There are two questions. One is what happened in Japan, right? So in Japan, the thing is very easy. They tell the mother not to give milk to the child. But they also tell that if you boil, the virus die. So you can boil the milk, the mother's milk, you can take out, you can boil, you can give the baby, no problem at all. You can do the other thing if you don't want to boil. You can freeze it and then thaw it. The virus is weak, it will die. Or inactivated, not die, truly speaking, inactivated. So in that way, you can give mother's milk to the child, but you can prevent the infection. Okay, the other was what happened to the group. You know, when we take NMRR, there are many restrictions about different, we cannot access to the IC card or something like this. So you are right, we want to do something, but we are unable at this moment to do anything. But we will talk with our counterpart in Sabah State Health Department, JKNS, to sort their advice and then do accordingly. Yeah.
Okay. Any other questions? I just know that for the mothers, you actually, that one is actually part of the preventions. Not can give the mix, but there are some steps to follow. Any other questions? So basically, it's actually the reason why you choose the 3 groups because of it's not really I mean purposely, right? Because you found the
purposely we did for the blood donors and the DBKK sample. But that was since we had it, we just give a try. Let's try what happened.
All the 3 group, which one is actually have the highest? I mean,
Bangi and Kudat,
okay.
more or less same.
Okay, any other questions? I think that's all for today. Thank you very much, Professor.
Okay. I thought you will ask me why male dominant.
It's very difficult.
when Iha show you the result, in Japan also female is dominant everywhere in the world. In Sabah, we find out male dominant. We don't know what is the reason. Our friend from Kudat area, Kudat Pitas, those area, they say they had a culture previously to give mother's milk to other child also, not own child, share with other child. So that may be one of the reason, and we have to find out actually, we don't know why male is predominant, whether they have multiple sexual partner or not, whether there were history of blood transfusion. We don't know because of the sampling, because the sampling was not done for HTLV-1, it was done for some other purpose. We think that's the reason, so we have to explore it in future.
Very interesting. It's quite different with Japan, right?
Yeah,
Japan.
Japan or other country. I think other countries also female more.
Ah.
So when there is sexual transmission, actually, from men to women, it goes easily than the other way. That's the reason why there is female predominance, one of the reasons. We don't know what is happening in Sabah. We have to do more studies.
Very interesting to know about that one. Okay, so I think there's no questions. We should end our sessions. To thank you very much, Professor Kamaruddin, as well as Associate Professor Iha Hidekatsu. Thank you for enlightening us with this HTLV type 1 related disease. And we hope that we can hear more, maybe after you got some finding after this. So thank you again. So sorry, we will continue with the quiz before we show the MMA CPD attendance as well as the feedback form. So the quiz is actually for the physical attendance only, okay, because the prize will be given here after this. So we will share the link as well as the code, but I'm so sorry for the online participants. This one is only for the physical participant only. There will be 5 prizes. Okay, you need to wait for a while. Get ready your phone. The fastest with the correct answer will be the winner. For the MMA CPD, please wait for a while. After the quiz, we will display for the CPD, MMA CPD. Okay, so you can click the start now. Kejap. Dia tak boleh. There's the code. You can start. Click start kan? Apa soalan dia tahu? Soalan voucher. Itulah. The link, the code. The pink, Kahoot pink. Dah bincang jawapan dulu. You can discuss for writing for that. But the apa? Yang paling cepat yang akan dapat hadiah. Kejap. There are some technical issue. Wait for the moment. Only 5 questions. Only 5 questions. Dia dah discuss dah. Okay, while waiting for the quiz, we show the MMA CPD first. Okay, maybe the online participants is eager to scan now. So we will share the MMA CPD first. But this one is scanned by normal QR code, right? Huh? Nak Kahoot dulu ke? Kahoot dulu. Okay, sorry. We go for the quiz first. Okay, so please enter the PIN code 3930879. Okay. For the physical participant only. You may scan or you may enter the game PIN. The winner is by questions. Sorry, not as a total. So 5 questions, 5 winners. Okay. So we can start now. Cepat. Okay, HTLV-1 belongs to the family Retroviridae, Coronaviridae, Filaviridae, not at all above. So it's actually Retroviridae. So who is the winner? I think it's total, right? It's a total, right? Okay, next. So, okay, so far AA is the leading. Okay, who is AA? Okay. You want to give now? Okay, next. Okay, we must remember you are the number one. Okay, number two questions. Next. Okay, HTLV-1 can be transmitted through airborne. True, false, not sure, maybe possible. Okay, no worries, this one not included in the exam marks. Dah jawab dah kot? All the... Okay, so the answer is false. So majority, 35 answer correct. So the leading is actually goes to R. Oh, Rizwan. Rizwan. Okay, we go to the question number 3. Ridwan. Bukan lah. Who asked Art? Okay. Kena minta nama. Okay, the third question, disease related to HTLV-1. So, severe acute respiratory infection, adult T-cell leukemia, lymphoma, cardiomyopathy, not sure. So the answer is adult T-cell leukemia, which is 39 correct. One got the wrong answer. Who is the leading now? Farah. Farah mana ni? Okay, Farah. Okay. So we go to the third, fourth questions. Number 4. What is the prevalence of HTLV-1 in Kudat, Sabah? 1.4, 2.4, 3.4, 4.4. Which one? Saya ingat, saya catat this one. Okay, 3.4. Okay, the answer is 3.4. Who is the leading now? IA. Who is IA? Okay, IA. So the last questions. In Kudat, Sabah, male are 2 times more likely to have HTLV-1 than female. True, false, not sure, maybe possible. This one should be majority correct. Okay, Prof kamu then highlighted just now. Okay, there's still wrong answer. Okay, 33 got true, but there's still a wrong answer here. It's okay. So now it's actually the final score, right? The first winner. Okay, the number 3, is it this one? D. Number 2, Sandakan. Number 1 is IA. Okay, maybe DIA, Sandakan, Ashdad, and M. The 5 people here. Okay, so we would like to invite Head of Department of Public Health, Professor Rosita, accompanied by Professor Rohaizat for a prize giving ceremony. Okay, Professor Rosita and Professor Rohaizat. So the first we would like is actually the first winner is actually IA. Who is IA? IA. Nama penuh apa? Izati. Okay, Izati. Congratulations. Okay, the second, Sandakan. Who is Sandakan? No Sandakan. Online ko. Can you boleh tengok dia punya full. So Sandakan tak ada. Tengok dia senarai pemenang tadi. Okay, maybe Sandakan online. So tak yang total. Dia boleh boleh tengok. D. Who is D? Dia Sandakan. Okay, D. Dina. D is Dina. D for Dina. Congratulations. Online. Congratulations, Dina. Okay. Okay, next. Tak, yang total punya jumlah tu. I rasa macam Ashdad kan? Ashdad tadi. Tapi dia keluar tadi. View summary. Kita tengok tak view summary tu? Ashdad, yes. Ashdad. Okay. Our MPA student, Ashdad. Congratulations, Ashdad. Okay. The next is the winner is M. Misra. Dah. Next. Ya, next apa? Mana menang? AA kot. Full report tu. Stop short je. Stop. Okay, so we give to AA. AA. Okay, AA. Okay, we give to AA. This one for quiz, not exam. Betul. AA. Okay, congratulations. Okay, so we will share the MMA CPD points for you to scan and give feedback for these symposiums. So before we end our sessions, I would like to pass back to Professor Rohaizat to give the end words from us. Thank you.
So thank you very much, Prof. Hasba. Right on time, I think we finished the session on time. First of all, I would like to apologize for all the hiccups. It happens, it happens in all online sessions. But I think overall, let's just give a big applause to our two speakers for their very interesting lectures for today. So, okay, you can scan the QR code and then fill up the feedback form, and then you get the free CPD points for MMA. Okay. So thank you very much, everyone, for attending this session today, and thank you very much again for Oita University for giving us opportunity to organize this and funding our. So this is like from the information just now, I think there's a lot of things to be studied, to be explored in this region on this HTLV-1. So I think these are good opportunities. We are calling for more interested person to join us aboard so that it's going to be a very good and beneficial research. We add up into the international data, to the regional and international data on what is going on these HTLV-related diseases. So I would like to thank all the organizing committee for their commitment, helping me for the past 1 month. And thanks all to all the attendees today, face-to-face and online. So with that, I think I will conclude the session today. Assalamualaikum warahmatullahi wabarakatuh. I think we shall take the opportunity to take the photo. Yeah. Okay. So how are we going to arrange? This Hassan, can you please help us? Untuk sesi bergambar, I think all of us boleh turun dan kita duduk sini in the background. Then, okay.